CD63 antigen is a protein that in humans is encoded by the CD63 gene. CD63 is mainly associated with membranes of intracellular vesicles, although cell surface expression may be induced. The protein encoded by this gene is a member of the transmembrane 4 superfamily, also known as the tetraspanin family. Most of these members are cell surface proteins that are characterized by the presence of 4 hydrophobic domains. The proteins mediate signal transduction events that play a role in the regulation of cell development, activation, growth, and motility. This encoded protein is a cell surface glycoprotein that is known to complex with integrins. It may function as a blood platelet activation marker. Deficiency of this protein is associated with Hermansky-Pudlak syndrome. Also, this gene has been associated with tumor progression. The use of alternate polyadenylation sites has been found for this gene. Alternative splicing results in multiple transcript variants encoding different proteins. CD63 is a good marker for flow cytometric quantification of in vitro activated basophils for diagnosis of Ig-mediated allergy. The test is commonly designated as basophil activation test. Initially, deletion and point mutants were used to investigate the role of the C-terminus, which contains a putative lysosomal targeting/internalization motif. C-terminal mutants showed increased surface expression and decreased intracellular localization relative to CD63WT. Antibody-induced internalization was reduced in C-terminal deletion mutants and abolished in GAR-A and URA mutants, showing the crucial role of these residues in internalization. CD63 is extensively and variably glycosylated, and the EC2 region contained 3 potential N-linked glycosylation sites. Mutants N130A and N150A were similar to HCD63WT with respect to intracellular localization and internalization. However, the HCD63 N172A mutant showed a mainly cell surface localization and low internalization. Expression of a mutant lacking all 3 glycosylation sites was very unstable. It was speculated that the reduced internalization of CD63 N172A might be due to changes in its interaction with cell surface molecules. Immunoprecipitation experiments showed some evidence of a protein associating with CD63 N172A, but this was not consistent. However, an association between CD63WT and I-circumflex superscript 22 integrin was shown by co-internalization of these proteins. Interactions with CD63 may therefore affect the trafficking and function of I-circumflex superscript 22 integrins. Recent work by other groups suggests that CD63 functions as an adapter, controlling the functions of interacting proteins such as integrins by inducing their internalization. Our findings indicate that CD63 glycosylation status can affect this role. A recent investigation showed that expression of CD63 positively correlates with the invasiveness of ovarian cancer. CD63 has been shown to interact with CD117 and CD82.